So our topic today is starting us along the path to cell-making proteins. Proteins do the work of the cell. Proteins are really where the interesting cell mechanisms happen. And we talked about, up until now, how does a protein structure allow its function? How are they regulated? What do they look like? And so now we're going to be talking about the process of transcription. What's this called overall?
Central dogma.
Central dogma that you're supposed to memorize. And the other part that you need to write on here and memorize is what the enzymes are that do these processes. So what's the one for today? Major enzyme of this day.
Polymerase.
RNA polymerase. Go ahead and write just RNA polymerase, because we're going to see that some RNAs actually function without being translated. And then the enzyme that does translation is the ribosome. So we're going to be starting today on this top half, looking at, all right, we've got DNA. If an organism contains a gene for a certain protein, how does it move it to the level of being transcribed into RNA? And the process of controlling transcription or regulating transcription can help a cell adjust its amount of protein later. So transcription is a very regulated process, and it can be fine-tuned. It's not just that every gene gets transcribed in the same speed, in the same manner. So at the DNA level, you've got a gene or you don't. If you're diploid, like we're talking about in this class, you've got probably 2 copies of it, which may or may not match. That's what you're talking about in genetics now. The copies match, the copies don't match. This book is going to consider one copy per gene. It leaves out diploid, recessive, dominant, and so on. So when it's talking about gene A, gene B, these are 2 different genes overall that encode different proteins. One of them could be transcribed a lot more than the other and make a lot more protein than the other. I'm going to put the screen up halfway here. And so one way to just control how much protein you make is how much transcription do you start. Do you start a lot of transcription, or do you start a little bit of transcription? And really the question here is, how often is a gene transcribed? How often is a gene transcribed? Another way of thinking about that is, when does the RNA polymerase bind there? So for this RNA transcript to get made in the first place, RNA polymerase has to land on the DNA, bind to it, and start transcribing. It's getting RNA polymerase started on the gene that really controls its transcription level. How often does RNA polymerase come and get started there? And the more often that RNA polymerase will land, so to speak, on a gene and transcribe it, the more messenger RNAs get made, the more proteins get made. So what were some of the DNA and RNA differences that you listed on your quiz? There was a quiz on this. On the quiz just now, what are some differences?
It's shorter. RNA is shorter.
RNA is shorter. I'm going to write that one up here, because that's usually true. But like when you go into molecular biology and you're working with artificial constructs, not always true. usually true. What else?
DNA needs a primer.
DNA needs a primer. I'm also going to write that outside the box. DNA, let's say construction, needs a primer.
RNA single stranded
DNA.
DNA.
RNA is usually single-stranded. DNA is double-stranded. So DNA is double-stranded. What are other huge fundamental differences between those two?
The base pair.
The base pairs, how so?
Well, the RNA has a, is it U instead of T?
Yeah, so they have different bases, which I'm just going to write like that.
RNAs have ribosome or ribonucleotide, and DNAs have deoxy.
I read yours. So talking about the backbones, ribose versus deoxyribose.
RNA has multiple functions, DNA functions, and storage.
RNA has multiple functions, which we're going to list a few of those later. DNA really is just the storage function. And those, any others? I'm going to write one more that is important for understanding the concept, but that books don't talk about as much. And that is that RNA is less stable than DNA. As in, a given RNA molecule, because it's single-stranded, because it doesn't have a binding partner, because it's shorter, it is more easily degraded in a cell. So RNA in general is much less stable than DNA. And that also reflects that DNA needs to be stable. It is the heritable storage for our genetic information. So most of those you probably knew already. It's worth reviewing here when we talk about how do we get from DNA instructions to an RNA transcript. Already got this. The other differences are ones that Dion was just referencing, is that RNA can have multiple functions, and most of those functions come from RNA that is folded up on itself, so base pairing with itself to have some regions that are double-stranded, or associating with an enzyme. So RNA can have multiple functions. And you might also want to write that RNA can base pair with itself. When RNA does base pair with itself within a single molecule, it will form regions of almost like helix that are fairly short, and then loops. And these are called stem loop structures. You ever heard that before? Where have you seen a stem loop structure before this class? You're going to read about them on Tuesday. So tRNAs look like a clover leaf with a lot of stem loop structures. And these base pairs operate in the same way as would DNA base pairs, really, except you have uracil instead of thymidine. this is a tRNA in a ribbon diagram. Okay, so some of the types of RNAs that can exist in a cell are messenger RNAs, and that's the main one that you tend to think about when you look at central dogma that code for proteins. That's the one I'm going to be talking about the most in this class. But it's really important to remember that there are other RNAs that function. Their function is to act as RNAs and not be translated into proteins. So all of these other groups, so I'll come over here and draw a bracket, these others, they're going to function in the cell as RNAs. Like RNA is the final destination for that gene, so to speak. Messenger RNAs eventually function as protein. So a messenger RNA on its own is not useful unless it's translated into protein. This is an intermediate step on the path to protein. But all of these others, once they reach the RNA stage, that's where they're going to operate. So if you print slides and write on them, however you connect one slide to a previous, like with a star or a number or whatever, if you go back to this level, all of the other RNAs that aren't messenger RNAs, when they reach this point, they're going to go on and do their function. They don't need to be translated. They're going to do something else as an RNA. That was something that I didn't get for a long time when I was younger. Oh yeah, I guess that wouldn't have to be translated to do its job. So those categories are ribosomal RNAs. These are ones that are parts of a ribosome. They help build the ribosome, which is a ginormous superstructure that we'll look at on Tuesday. Transfer RNAs bring amino acids to the ribosome. I like that definition more than serving as adapters during protein synthesis. Transfer RNAs bring amino acids to the ribosome for use in translation. These other two groups are somewhat more recently discovered. So microRNAs, you may read about those in one of your papers. You may learn about it in genetics or molecular biology. They're involved in regulating transcription. I would write out there, regulate transcription. And then we have the class of other non-coding, which do a lot of different things. I'm not going to go through those specifically, but not every RNA has as its fate that it's going to get translated into protein. Likewise, for different sets of these RNAs, they're being made by different RNA polymerases. So in eukaryotes, like us, there are 3 varieties of RNA polymerases, which we call Roman numeral 1, Roman numeral 2, and Roman numeral 3. The one that translates messenger RNAs is RNA polymerase 2, and that fact is worth remembering. That one comes up on major standardized tests sometime. Maybe because it's easy to write that question. RNA polymerase 2 transcribes protein coding genes. The other RNA polymerases share between them the jobs of transcribing those other RNAs. So you need RNA polymerase 3 and RNA polymerase 1 to build ribosomal structural transcripts and other things.
Was it 2 that you said?
RNA polymerase 2 is the one that transcribes messenger RNAs, yeah. And for this class, that's the one you need to memorize its major function. So, most of the time from now, I'm going to be talking about how do messenger RNAs get transcribed and then translated. And at the beginning of that process, the information that's needed to make this transcript, this RNA transcript, is in the DNA. DNA is double-stranded, RNA is single-stranded. RNA polymerase, no matter what kind it is, can only read one strand of DNA. It can only read one thing at a time. It can only, in essence, see one side of the double helix when it's operating. So it's going to read one strand of DNA, and it's going to use that information to produce a single-stranded RNA transcript. Okay, so here's a question I want to ask maybe a set of you who have not had molecular biology yet. See if you remember this. If this is the RNA that is produced during transcription, well, it's on this, I hate this, it's on the screen. Okay, this is the RNA that is produced. It has the same nucleotide sequence as the coding strand, except there would be T to U changes. You see how that is? This has the same sequence as the coding strand. Is that a new concept? Have you heard of coding and template? Iffy? So what I want you to put on here is that this is the strand, the template strand is the one that RNA polymerase actually reads. RNA polymerase reads, scans the template strand, and that's where it's getting its instructions. So it's going to be scanning here and using this strand with complementary base pairing rules to produce this messenger RNA. It reads the template. It reads the template strand. It produces, in essence, a copy of what we call the coding strand. So on any given DNA, let's see, I need white space for this. Hang on just a blank slide. Any given DNA that we're looking at will always be double-stranded. And if we're looking in the region where there's a gene, this is strand 1, strand 2, there is not a consistent side that contains genes. So if you imagine this DNA being extremely long, there might be one gene that is using that as the coding strand, that half. There might be another gene that's using this as the coding strand. There's two options for where a gene could be. A lot of viruses actually use both strands for genes as an efficiency mechanism for packing a lot of genes into a small space. But in eukaryotes, it's usually one side or the other. So RNA polymerase can only read one strand. And which strand that could be is different for different parts of the DNA for different genes. Is that making sense, what I'm trying to say? Maybe it imagine that you're an RNA polymerase. Where are you in the cell if you're going to be doing your job right now? You should be in the nucleus where the DNA is. RNA polymerase, the major pool of it exists in the nucleus. And RNA polymerase, its main job is to bind to DNA, open the helix, and start transcribing off of the template strand. It will start transcribing, in essence, reading the template strand, allowing base pairing of new floating RNA nucleotides that are floating around in a pool in the nucleus. And they will be joined into a new messenger RNA transcript. So several important things about this slide. RNA polymerase acts in the nucleus. There's a pool of it there. There's a lot of it. There's a lot of RNA polymerase in the nucleus. It may or may not be bound to DNA at any given time. There's plenty of it. There are also 4 pools of RNA nucleotides. Again, in incredible numbers that exist in the nucleus. And so you would have U, G, A, and C varieties of those floating around in here. The third important factor is that the transcript that gets made is built 5' to 3'. The messenger RNA is built starting with the 5' end and finishing eventually at the 3' end. How do we specify ends of nucleic acids? Where does that come from? By the carbon number. This is where it's going back to. It is related to the carbon number of the sugar that the end molecule is attached to. So the 5' end has a phosphate group that is attached to carbon 5. The 3' end is the other end. 3' end down here, if this is the last base in the chain, that would actually be a free OH group down there. So the 5' end is going to have a phosphate. The 3' end, if it's the real end, has a free OH group, hydroxyl group at the end.
So is the template strand always on the 5'? Or is it always going to attach to that one?
So the question is the template strand always 3' to 5'? What I would say is the template strand is read 3' to 5'. The template strand always exists, just sitting there. But when a ribosome is reading it, a polymerase scans the template strand from 3' to 5' direction. And when it's doing that, it's building the new messenger RNA to be like the coding strand, which is running in the 5' to 3' direction. So that's how transcription is generally operating. I want to talk about the concept here of how these come in here. There's a pool of ribonucleoside triphosphates, GUAC. They're floating around. They don't line up in a line in order and come in here. They're bouncing in and out of here. And if they happen to form the correct base pair, they stay long enough to be put into the chain. We're going to see that on Tuesday, a similar concept with how ribosomes operate. But remember, this is all operating in a diffusion, jostling, crowded, mosh pit environment of molecules. So this is generally how transcription operates. But it doesn't get started until RNA polymerase is bound to the correct place on the DNA. RNA polymerase has to find a place to start, bind there, and then get going. And the decision, what I call a decision, whether to bind, whether to start, that's really controlling. Are we going to make messenger RNA, or are we going to make protein later? So how does RNA decide that? These structures are called bottle brush structures. Bottle brush, it's really one word. Bottle brush structures. And they are depicting some very popular genes being transcribed. And by popular, I mean there's something about these genes that is great at attracting RNA polymerase to come there and transcribe them. These are ribosomal RNA genes. And you can see a little bit of the same type of depiction up here. This actually loops around, and it's a segment of the genome that contains repeated copies of ribosomal RNA genes. So this is an example. You've got more than 2 copies of ribosomal RNA genes because you need more than that to make enough. So you wouldn't just have 2 copies, you've got a lot of copies. And they're often lined up together in the DNA. This is showing a gene that is so popular, that is so attractive to RNA polymerase, that RNA polymerases can't wait to get on there. It's like the good Dollywood rides. Like, people are lining up. As soon as you can get on it, you get on it. So what you're looking at here are RNA messenger or RNA transcripts being produced, and you can imagine the polymerases coming along in a chain, and they're all transcribing at the same time. Something about these genes is so popular, is so attractive to polymerases that they transcribe like crazy. But not every gene acts this way. Some genes are unpopular. What's the difference? It ends up being regulation. What would make a gene attractive to RNA polymerase or not attractive?
Their body needs them more.
Because our body needs these genes more, they have been selected to be more attractive. So it's like the question is, if I'm a gene, what could I have that would make me more attractive or more popular to RNA polymerase? What RNA polymerases really look for in a place to move to land on, to transcribe, we tend to call that the promoter. The promoter. The promoter is really where RNA polymerase goes to get started in its work. Have you heard of promoter? Maybe? I'm sure that if you have looked at a promoter or learned about promoters before, that they have a definition that is written down in a book. I want you to think about promoter as a landing spot for RNA polymerase and some other associated proteins. A promoter is a place that RNA polymerase will come to and bind to prepare for starting to transcribe. The other thing to note is, okay, it's a landing spot for RNA polymerase and other associated proteins. And a lot of times we call those proteins transcription factors.
Bless you.
So I'm writing over here, those other proteins are transcription factors. So then that begs the question, well, okay, promoter is a landing spot. What is a promoter? A promoter picture is showing it as being next to the gene. Really, a promoter is part of the gene. So promoter will be nearby, at the very least, to a gene. It should be considered as part of it. Most of the time, it's upstream. Upstream. Or off to one side of the gene. And the promoter is not in the protein coding region. Or the promoter is a little bit distance away than the part of the gene that is going to be important for the function of the transcript. It's a landing spot. It's nearby. It's like those lights at the airport that tell the plane, no, you're really supposed to land over here. Please land here. That's kind of what a promoter is like. So promoters also give RNA polymerase information about which direction it ought to be going, so to speak. They will be associated with genes. So if RNA polymerase lands here and it makes this transcript, this messenger RNA, which one of these was the coding strand?
The 5' to 3'.
If this is 5' to 3', which it would be because messenger RNA has to be built that way, this is the coding strand, this was the template strand. So this one is what RNA polymerase really would read. On this one over here, so if that was confusing, this is RNA transcript. It's showing that it started being made here. It's proceeding in this direction. It had to be started built from the 5' end to the 3' end. For this gene, which one is the coding strand? It would be the one that's running 5' to 3', which would be the bottom. Top and bottom don't really exist in a cell. The bottom, this one would be the template strand. This one would be where RNA polymerase gets started. So on this one, this is the template strand. For gene B, this is the template strand. So you can split DNA in half and make a new copy. That's replication. That's heritable information. You do need both strands in a functional situation. Yes.
So since it's unzipped, can multiple RNA polymerases transcribe at the same time? Like, would there be two polymerases if they wanted to transcribe both those genes? Can they do that at the same time?
So the question is, if this unzips, can two polymerases go at the same time? Is that yes?
Okay.
Or 50, or whatever is shown in this. Yes. If the promoter is attractive enough to get them to come over that often, yes. Yeah. Okay. So RNA polymerase, if it can be attracted to a promoter, it can get started. It can transcribe using the template strand to make a messenger RNA or other RNA that is sequence equivalent to the coding strand. So here it is starting now. RNA polymerase is the light blue blob. These other factors, we do not learn their specific functions in this class. So don't freak out about this picture for learning that the telephone is TF2B and so on. I don't know. That is based on its real shape. But this picture, what I want you to know about it is looking at how many factors it takes to get RNA polymerase to get started. RNA polymerase itself, I think about it as like a spastic little toddler who's supposed to be coloring right now. If only you could get them started coloring and be like, can you color? It's your favorite. It's Lightning McQueen right here. Could you come over here? Could you help me get him over here and color? So once RNA polymerase gets started, it's gonna color, it's gonna transcribe. It takes a village literally to get it to come over there, sit down, focus, so to speak, start transcribing. And that's what transcription factors are for. So all of these other blobs that are not RNA polymerase 2, these are transcription factors. And they're going to bind sequentially to the promoter, to the DNA, and their combined binding surface. So transcription factors is combined let's see, I want to make it say something different. Their combined 3D surface is what binds RNA polymerase. Really, it's like RNA polymerase doesn't look for DNA, it looks for transcription factors blobbed up in a group. You can, maybe RNA polymerase is like a rubbernecker or a car driving down the highway, and if like suddenly there's a lot of people gathered over there, like, oh, what's going on here? There's a lot of binding surface for it is the assemblage of these transcription factors. So the transcription factors are the ones that are binding DNA. One reason why there are so many ones of them is that the more individuals you have involved in the regulation, the more fine-tuned the regulation. The more you get bound, the stronger RNA polymerase will be recruited to the promoter, the more it will transcribe. So it's the transcription factors that are binding the DNA. The RNA polymerase is then binding the transcription factors to be recruited to the promoter. So this picture is showing the first transcription factor on the scene of the DNA. And this one is called TATA binding protein. So here it's showing the protein. This is a dimer. No, it's not. It's not a dimer. It's not a dimer. It is one continuous backbone. There's the N-terminus, there's the C-terminus. But in essence, it sort of acts like a dimer because the blue part of it is a reverse flipped mirror image of the green part of it. They have similar shapes between those parts. And this protein binds to this sequence. TATAAAAG. It binds to that sequence. The factors that are involved in making the protein bind to the sequence are the same things that make any protein bind to any protein. Same concepts for protein-protein binding are controlling protein DNA binding. So what factors are involved in that? Thinking back to protein function.
Electrostatic surface match.
The electrostatic surface match between these, the shape, so on. DNA sequences, or let's say nucleotide sequences, nucleotide sequences have electrostatic surfaces of their own. they have electron clouds with, partial polarities, opportunities for hydrogen bonding. They are a binding surface too. They're a binding surface too. So this sequence corresponds with the binding surface of this TATA binding protein. It's called TATA because TATA is in the sequence. This also, people refer to it as the core promoter sequence. Every promoter in a eukaryote needs to have that sequence. TATA. AG. Say that again. It is a bunch of A's and a bunch of T's, but it's not that you need 4 A's, 2 T's, and a G. It has to be in that sequence. Has to be in that sequence. This is the core sequence. This is representing what it takes to get the first transcription factor, which is TATA binding protein, connected to this larger one, to come bind here. Other sequences in this region might be recruiting different transcription factors that together can attract the attention of RNA polymerase and get it to come over. So I tend to talk about weak and strong promoters. So the promoter is what gets RNA polymerase eventually to come over. And promoters have different sequences. Promoters have different strengths. All promoters need that core TATA AAG sequence. But the other sequences around them controls what else binds. So a strong promoter will be one that attracts RNA polymerase often. And a strong promoter would have this core promoter sequence plus others around it. A weak promoter attracts RNA polymerase less often. Less often. And that's sequence based. In genetic engineering, or when people go and create new versions of a gene, a lot of times you can cut and paste the business part of the gene with the promoter. You can put a gene behind a strong promoter and get it made a lot. Likewise, if there's a mutation in the promoter, that could make things bind better, it could make things bind worse. You're changing the sequence, you're changing the surface for binding proteins. Yes?
So what makes it stronger? Just
What makes it stronger? Does it have the sequence to bind the most attractive set of proteins
so like
for RNA
the kind of
polymerase?
sequences make
The
it stronger?
sequence, yeah, the sequence makes it stronger. And the sequence for a strong promoter in a bacterium is not the same as a sequence for a strong promoter for us. I don't know actually what time it is, but let's have a break here.